Hi to everyone. So I'm going to speak about neuroimaging at Croatian Institute for Brain Research. And we have been using neuroimaging at our institution for more than 20 years. So from the beginning, we realized this is really excellent tool if you want to study morphology of the brain at the level of whole brain. But if you want to go to details, then you need to find another tool. And hopefully for us, we already had this precise tool like histology, immunocytochemistry, and all other kind of staining. So basically combination of these different tools lead us to some significant, we believe significant scientific papers. And as I could see from the Highball project, you are doing something very similar, but just on amazingly advanced technical level. So when we speak about the brain, at least at Croatian Institute for Brain Research, we think that you cannot understand the structure and function of adult brain if you are not familiar with the perinatal brain development. So this could be one of the messages. And if you want to understand perinatal brain development, then we believe the main structures are actually transient fetal structures, like subplate zone, like crossroads of white matter, and a sagittal strata. So these are zones which are some kind of focus of our scientific interest. And I will show you some of our previous research first, and then I will try to explain the concept of radial vulnerability, which is some kind of connection between basic science and clinical practice. And also I'm going to say something about our ongoing research, which combine all this previous work at our institute with intention to find the perinatal biomarkers of normal and abnormal brain development. So one idea was to combine neuroimaging with histology to realize what actually this neuroimaging means. And another idea and another goal is to combine neuroimaging with clinical outcome in children after perinatal brain lesion to see how this could be applied to improve diagnostics and therapy for these kids. So first I'm going to say something about subplate zone. And this is a really difficult task if you have to speak about subplate after Professor Kostovic, but I will try to do my best. So when we started 20 years ago, everyone at Institute were very happy because when you make MRI of the perinatal brain, then most of that perinatal brain are actually subplate. This is the most voluminous part of perinatal brain during some phases of gestation. So when you check to the cerebral wall, you could see that there is some kind of different signal intensities through the cerebral wall. But if you want to realize in more detail what that changes of signal intensity means, then this could be a tricky job if you just look to neuroimaging. But if you help yourself with other techniques, then things become a little bit clarified. So for example, if you use this crescil violet staining, then you could get all the layers of cortical wall from the ventricular zone, subventricular zone, intermediate zone, subplate zone, cortical plate, and marginal zone, even fiber-rich zone within subventricular zone. So this was some kind of help, because when you combine histology and neuroimaging, then somehow you are able to realize what that signal intensity and how this signal intensity could be correlated with cells, fibers, and extracellular matrix. So this was at the beginning, and this paper actually was published in many textbooks. Another thing which was already been said today is that subplate zone is not something restricted to fetal development, but you could find subplate even after the birth. So for example, if you look to term newborn, you should find some difference of signal intensity below the cortex, and especially in the apical part of gyruses. And when you correlate this finding to histological section, you will see that this is actually remnant of the subplate. And this remnant of the subplate is very important because it's differently distributed through the brain. So it's different in primary motor and sensory area than in, let's say, prefrontal associative cortex. So developmental changes of subplate are a good way to study maturation of the brain. And if I could put all together, this could be some kind of dynamic that you could easily realize. So for example, if you start to analyze the brain at 24 weeks after the birth, then party is already over if we speak about subplate zone, because you will not find any subplate zone at that time. But if you start at 24 weeks of gestation, this is completely different because the subplate zone is the main player in cerebral wall. So during the time, the volume of subplate will slowly decrease, but it is important to notice that even after birth, you will find subplate in the brain. And even if you take a look at the brain of 1-year-old child, you still will find some subplate remnant at the top of gyruses below the cortical plate. So this is important to know. So what could be the function of such a voluminous part of cerebral wall during development? I will try to simplify this just by making simple analogy between architecture and cytoarchitecture. So if you just switch from the neuroscience to architecture for a second, you would realize then when you are making some simple structure, for example, like this brick wall, then you actually don't need elaborating scaffolding. You could use some improvisation or some simple scaffolding, and every big brick could be placed on a proper position. But if you are making some more complex building, then you need to have complex scaffolding. So it's very similar with the brain. When you have a rodent brain, as Professor Judas said 2 days ago, you have subplate just like tiny line. But if you go to study monkey brains or humans brains, then this complex structure need complex scaffolding for development. So just by looking to the subplate zone in humans, you could realize that human brain is the most complex of all brain. This is one analogy. Another analogy is when a building is finished, you will remove your scaffolding. So if you come after 10 years, you could find very mysterious how people put the ringing bell on the top of the church. But if you know how that looks like during construction, then this mystery will not be so problematic. So it's very similar with the brain. And finally, this is different, and analogy doesn't work in this case, but subplate zone is not only important for structural organization. Basically, subplate zone is also important for functional maturation. As Professor Kostovic said, these neurons are some kind of amplifier for thalamo-cortical or long associative connection to the subplate. So this is some simplification of the role of the subplate, and the message is you cannot have complex brain without complex scaffolding or without subplate. So let me say something about crossroads of white matter. This is another spot of interest for us, because during our histological studies, we realized that there are some locations inside white matter which are completely different when you check for the extracellular matrix, for axonal guidance molecules, for the fiber orientation. And actually these are known places of crossing of different kind of fibers. And this is something which is really important from the perspective of clinical neuroradiology, because if you have lesion in this part of the brain, then clinical outcome could affect different systems. So you could have sensory, motor, or cognitive deficit just by lesion in a very tiny location of the brain. And you would need to believe me, but in reality, it could be seen more clearly than on this slide. This crossroad region could easily be seen in a premature brain. And for example, if you take image in 27 weeks of gestation, then you could easily see in this frontal crossroad region. But if you take this image in 34 weeks of gestation, visibility of this crossroad region is decreased. And then if you take image at term equivalent age, you still could see the crossroad region. But if you take image at 2 years, then this crossroad region is visible no more. So it is important to see how clinically you could use visibility of this crossroad region. Some people claimed before that this is pathological sign, but basically we believe that visibility of crossroad region actually is good marker of normal development. And when you clearly could see borders of crossroad region, that usually means that outcome for this kid will be good. And there are numerous papers which shows exactly that. But there is also some kind of frustration in pediatric neuroradiology, because it's a big problem to correlate a lesion with clinical outcomes. So Professor Kostovic developed a concept of radial vulnerability, which is template or framework when you think about correlation of lesion and clinical outcome. And just to explain this concept, I will briefly say that when you have extremely early premature baby, then usually you have problem close to ventricular, in paraventricular region. If you have late preterm baby, then location of the lesion is moving to the centrum semiovale. And if you have perinatal asphyxia at term, then usually cortical and subcortical regions will be affected. So there is some kind of movement of lesions from the ventricle to cortical direction. And another thing is that during brain development, there is precise timetable of growing of different classes of axonal pathways. And there is also very precise spatial organization and position of all these axonal pathways. And the crucial thing is that these axonal pathways are most vulnerable to the pathogenic factors during the period of most intensive growth. So these are the foundation of this radial vulnerability concept. And this vulnerability of immature pathways could easily be presented like some other situation from real life. So during development, when you have immature system, he is very sensitive to some pathogenic factors, but the good thing that vulnerability is the biggest, but also brain plasticity is the biggest during that period of time. So combining all this together, we came to the logical concept that if you have lesion at certain time during fetal development, that will affect one part of cerebral wall closer or more distance from a ventricular area. And this specific location will damage specific axonal pathways. And damage of specific axonal pathways could be correlated with clinical outcome. It sounds very logical, but in practice, it is still very complicated to correlate a lesion with types of axonal pathways affected and with possible clinical outcome. And another thing which is problematic is that sometimes you have completely normal brain and still you have kids with a disability. So even if, let's say, 7 or up to 10% of cerebral palsy, you will have completely normal structural brain. So this is our concept. And finally, I came to the sagittal strata, which is another structure that we believe could be very useful in a clinical practice, because this sagittal strata, at least it seems from a few of our works, is a good biomarker of normal and abnormal development. Visibility of sagittal strata is high in premature babies. So if you make scanning at a premature baby of 30 weeks, you would see this sagittal strata around trigone or occipital horns of lateral ventricles. And when you okay, can I go? Okay. When you make scanning at term equivalent age, then you have reduced visibility of sagittal strata, but it is not the same reduction when you speak, for example, of premature baby, or when you speak of term child. And visibility is usually reduced in premature babies. So somehow it seems that this reduced visibility is a biomarker of some developmental impairments. And we noticed, for example, when we have twins born at the same day, that one with less visible sagittal strata will have worst clinical outcome, while another with clearly visible sagittal strata will have good clinical outcome. And that also works for extremely early preterm babies when you expect some kind of clinical complication. But in this case, with really very well visible sagittal strata, even this extremely preterm baby was very well after neurological and psychological testing. Of course, you cannot conclude anything from 2 kids, but we have cohort of 70 kids, and that really works, and this sagittal strata is good prognostic marker or later development. Just by looking to neuroimaging, you cannot realize what you are actually looking at, because resolution is not good enough to see all the details. But when you make this histological section on an insulin sagittal strata, then you see extremely complex laminar organization of cell fibers. And we actually believe that during premature birth, you have damage of the pre-oligodendrocytes, and because of that damage of pre-oligodendrocytes, you will later have a reduced myelination in this region, and because of the reduced myelination, you will have reduced visibility of sagittal strata. So when we made the histological staining, then you see in a normal term baby, you easily see these cell lines which are missing in a premature baby. And when we checked the myelination with myelin-based protein immunocytochemistry, then in a normal baby you see this myelin basic protein, but in this premature baby, this myelin is missing. So this could be explanation of the neuroimaging findings, but also it could be explanation why these neurological impairments appear. This year we published one paper which correlates visibility of sagittal strata with neonatal visual assessment score, and it was shown that visibility of sagittal strata actually is a strong predictor of total score on this test. So this is interesting because this is something that you could analyze on classical structural MRI slices of the fetus. And our idea is to bring this to neuroradiological community, to people take care about what is visibility of sagittal strata, because we think that this is quite useful parameter. So just shortly, I will say what we are doing at the moment. So as I said, we are trying to combine neuroimaging with regular clinical practice, and special focus for us are these premature babies. So in Croatia, every year there is something like 2,000 premature babies. 6% of all births are premature babies, and in most of the countries, this percentage goes from 5 to 15. So this is huge number of babies, and many of these babies will have some kind of neurological problem during development, and it's always useful if you could recognize that even before onset of sensory motor or cognitive deficit. So our idea is to take 2 MRI exams, first immediately after the premature birth, and the second one at term equivalent age, and also to combine these findings with a set of pediatric testing starting at immediately after the birth up to third year of age. So idea is that by analyzing this term equivalent age MRI examination, first make original MRI scoring system, analyzing visibility of some structure like crossroads of white matter, sagittal strata, and so on, and trying to correlate visibility of these structures with clinical outcome. Another approach is to make this diffusion imaging, especially adjusted for neonatal brain, and then to precisely define which kind of axonal pathways are affected by premature birth, and also to make cortical morphometry to see if this data of cortical structure somehow could be correlated with clinical outcome. So the objective of our ongoing research is to define structural and diffusion biomarkers of normal and abnormal perinatal brain development, with the idea that this is really useful for these premature babies, because in that way you could start early therapeutics, and it was shown that if you start early, the final clinical outcome in this kid is then much better. Okay, thank you very much.